So I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation. And I pay my respects to their elders past and present, and also elders from any other community, any members of any community that might be here today. So I'd like to introduce Professor Ed Stanley. So he actually completed his PhD here in Melbourne at the Ludwig, and then he moved away to London for a postdoc. He came back to Australia and actually joined WEHI, the lab of Richard Harvey. And then about 15 years ago, he moved to Monash, where with his collaborator, he started his own group. And he's most recently at the MCRI at Murdoch. So throughout his whole career, he's focused on pluripotency, differentiation, and development. And it's the utility of those things that he's going to tell us about today. Thanks.
Okay, well, thanks very much for asking me to come along and talk to you today. I realised the title that I'd been given to talk on, and quite frankly I couldn't be bothered changing it, is not really true, and that is that you can't really use human pluripotent stem cells to model human development. In fact, you can use mouse development to model human pluripotent stem cell development in vitro, but extrapolating to human development has a whole lot of issues attached to it, which I hope during this talk I'll be able to give you some flavour of. So one of the things you'll get from the talk is that we use developmental biology information to try and direct our pluripotent stem cells towards cell types that we're interested in. And I'll go through a whole host of different cell types today that over the years we've been involved in generating. But the information that you have is very dependent on where you start from. So I was reminded of this as I was trying to enter this building today. And I read this sign and it says, follow the blue painted footprints and the arrows. Now, of course, I'd come from the wrong direction. There were no footprints and there were no arrows. So following the directions of developmental biology can be fraught depending on where you start from. So I'm going to give you a few slides about stem cells, and I hope most of you are familiar enough with this area. So traditionally, stem cells designed as a cell that can renew itself, that can differentiate into a more mature cell type, and people believe that in the adult at least, stem cells living in tissues are there to help repair those tissues after injury or just life in general. There are two main types of stem cells that people talk about. There are the pluripotent stem cells, which is going to be really all I'll talk about today. And they have a particular property, and that is that you can grow them for a very long time in the lab and they still retain their stem cell characteristics. They're relatively easy to grow, and this is one of the reasons people work on them. They can turn into any cell type. Now, in the case of mouse embryonic stem cells, for example, this has been experimentally proven. In the case of human pluripotent stem cells, it's actually assumed, and when people have tried to turn their stem cells into a cell type they desire, generally they've been reasonably successful. So the two types of pluripotent stem cells people talk about are the embryonic stem cells, which are made, in the case of humans, from a 6-day-old embryo, a blastocyst, and induced pluripotent stem cells that are made by reprogramming adult cells. And I'll just briefly run over those in a minute. So the adult stem cells, historically, they've been difficult to grow in the laboratory, and although people have got quite good at growing things out of liver or gut, for example, growing a haematopoietic stem cell, which I'll touch on towards the end of this talk, is still something that really can't be done in any reliable fashion. They're purified from adult tissues and they tend to make a restricted range of cell types. And there was a lot of kerfuffle probably around 10 or 15 years ago where people hoped that adult stem cells could turn into everything, but I think that kerfuffle has died down, and now people appreciate that haematopoietic stem cells, for example, make blood cells. Mesenchymal stem cells make things like bone, fat and cartilage. So the stem cells in your tissues tend to make what they need to make in those tissues. I guess the important thing is that we do know a lot about haematopoietic stem cells because we know that in the case of a human, they can be used clinically in the form of a bone marrow transplant for leukemias. So as I mentioned, embryonic stem cells come from a blastocyst. There's a picture of one. The red stuff in the middle there is what we call the inner cell mass. And if in the old days you would grow those cells on a layer of fibroblast feeder cells, what you see there is a colony of human ES cells with maybe 10,000 cells in it. And they pack together as a tight epithelium. Induced pluripotent stem cells are pretty much the same but different. So you'll be aware more than 10 years ago now, Shinya Yamanaka, Japanese scientist, figured out how to take an adult cell like a fibroblast and turn that into something that for all intents and purposes looks like an embryonic stem cell. And so it grows as a tightly packed epithelial cell, and it is made by applying either genes or chemicals or combinations of both to an adult cell to transform its epigenetic landscape into something that looks like an embryonic stem cell. So the use of these cells, which I'm not really going to go into today, mainly because it's a very big area, is that those cells are a genetic match to the people that you make them from. And so if that person has a disease or a genetic condition, this is a method where you can capture that genetic information in an immortal cell line, and then you can differentiate those cells into the cell type that might experience the disease. And thus, people are using that kind of thing for disease modelling. In fact, that's something we do a lot of. So this is the kind of slide that maybe Jamie Thomson would have put up 20 years ago when he first isolated human embryonic stem cells. If you could make a whole range of different cell types from an embryonic stem cell, then maybe you could do a whole lot of things with those. You could, for example, come up with cell replacement therapies for a whole lot of diseases that, for at the moment, there aren't really any viable options. And this is starting to happen. It's taken a long time, but there are clinical trials now where people are looking at in vitro derived human cell types made from, in the main, embryonic stem cells for the purposes of treating a whole range of different conditions. Perhaps one of the ones I find interesting, because it's closer to an area I'm working on, is the derivation of insulin-producing beta cells for the purposes of treating type 1 diabetes. But something we all realised when we started working on this area is that the big gains to be made in this experimental approach was to use the cells you made for looking at drug screening, disease modelling, and all the kind of boring stuff that the media is not really interested in. And these kinds of things are going on as well, particularly in the heart. So if you can make human heart cells, they're a very good substrate for screening drugs against. And one of the reasons they are useful is because the human heart and its ion channels and its behaviour is so much different from any other species. If you want to look to see if your drug has cardiotoxicity or any other impacts on the cardiac physiology, then a human pluripotent stem cell-derived cardiomyocyte is really by far the best option. So I'll just go through briefly some of the methods that we use in the lab and some of the techniques and the thinking behind what we do. So before you can use stem cells to try and understand disease and development, there's a few things you need to be able to do. You need to be able to identify the cell types you're looking for. And we use lineage-specific reporters. So if any of you have looked at embryology or developmental biology, or even, for example, pathology, you know that if you take a bit of brain tissue, you've taken it from a brain, and it's going to be brain tissue. But if you've got a dish full of amorphous cells that all look the same, how do you know the cell that you're interested in is in amongst that? Well, what we do is we say, okay, we know this gene marks a particular cell type. We genetically modify our starting cells such that when those cells differentiate into the cell type we're interested in, they switch on the gene, which switches on a reporter, and we can see a green cell or a red cell or a blue cell. That, from a technical point of view, makes it a lot easier to follow what's going on. The other thing, and this is harking back to my comment about the ability to use differentiation as a way of modelling human development, it's critical that you need to have methods for generating cells. And what that means for most labs these days is you need to have chemically defined differentiation media. You need to know what you're putting into the system if you're going to try and control it. What that tells you is that what you put into the system dictates the outcomes, and this is why it's really problematic to consider this system as a model for human development or differentiation, because it's artificial. You are the person that dictates which way cells differentiate, what they can become or can't become. So to walk back from that and say, I'm modelling human development is a little bit contrived. Okay, so we use a range of different protocols for making the stuff we're interested in. We use what's called an embryoid body differentiation method sometimes, and those embryoid bodies are clusters of pluripotent stem cells, like a little sphere, and I'll show you some pictures soon. The good thing about those is that they have a 3D environment, and a lot of cells like to be in a 3D environment. It's part of their physiology, if you like. So they can differentiate in a way that they prefer. But under some circumstances, it's nice to have more control over what you're letting the cells do and see, and people use, and we do, flat cultures for these. So that's a monolayer of cells growing on a flask. Most of our methods, however, use combinations of these two, switching from one to the other, depending on what stage of what we're looking at. And as I said, we use fluorescent reporter genes to find cells we're interested in. So this is a picture from my long-term collaborators, Andrew Alifanti and Elizabeth Ng. And what you're seeing here are some green blood cells, and they're green because GFP is being expressed from a haematopoietic specific transcription factor, RUNX1C. And the red is an endothelial cell type, in this case a haemogenic endothelial cell type marked by expression of SOX17. And what we've done is we've inserted an M-cherry reporter into the SOX17 locus. So this cell line goes red when it's making vascular structures, and then as that vascular structure starts turning into blood, we get induction of green. So what that means is we can look down the microscope and see things happening, and that's important if you're trying to optimise differentiation protocols. What I'm going to go briefly into now is a method that we developed more than 10 years ago, and we don't tend to use it that much anymore, but it's still useful in terms of a teaching seminar. So this is what we call the spin embryoid body method. It's a method for taking pluripotent stem cells, dissociating them to a single cell solution, and then putting them into a 96-well tray, and then spinning those cells down such that they aggregate. Now, this had two effects. One is that it rescued the survival of the cells because they're an epithelial cell and they don't like being apart. But it also generated a uniform size of cells in each well, and this was very useful for determining differentiation trajectories. And you can see that in this series of pictures here how we could adjust the size of the embryoid body across simply by dialing up the number of cells going into it. This is important because the embryoid body size in those days dictates the direction of differentiation, and because your cells are talking to each other, if you want to put in factors to override that, then having an idea of how many cells you've got in there is important. Okay. So I think what I've done next is I've got a whole list of things which I think are take-home messages from this talk, and I'll hopefully have examples of each of them as I go through. The first thing is that the first decisions are critical in differentiation. The direction of differentiation is by and large determined in the first 24 hours, and it's not reversible. So if you stuff that up, and you're trying to do a 6-week differentiation, then you can't go back from that position. So you can imagine driving from Melbourne to Sydney, and if you're heading off to Geelong, you're never going to get there. So the other thing is that differentiation appears to proceed stepwise through obligate intermediates. So we know this from embryology, that cells, as they move from one state to the next, there are certain checkpoints they have to go through, and during differentiation in vitro, we try to see those step points happening because that tells us we're on the right track. This is a little bit contentious, but it's an idea that's been pushed mainly by Kyle Lowe, who's currently working in Stanford, and that is that most of the decisions can be broken down into binary decisions between different differentiation outcomes. And I'll hopefully give you some examples of that. A consequence of this, and I'll show you examples again soon, that growth factors and signalling pathways do two things. They promote a differentiation outcome, but they also block outcomes. And in terms of trying to push your cells towards a cell type that you're interested in, it's not enough to just give them the factors they want. The other thing that you have to do, and I think this is a great concept for a lot of different experimental approaches, but the other thing you have to do is you have to stop them from taking the alternatives. And so most protocols that we use these days have two components to them. They have an inducing component, that is pushing the cell in the direction you want, and they'll simultaneously have blocking components to try and stop the outcomes that you don't want to happen from happening. The other thing that we see quite commonly is once a decision point is reached, like once a cell has become a particular intermediate, then a whole lot of new options come up that are available. And so in terms of a differentiation idea, what you do is you push the cells to the first step, blocking the alternatives. Once they've reached that step, they have a whole new range of options available to them. And so for you to go to the next step, you have to do that again, push them again, but block all those options. What you'll see, and if any of you have studied developmental biology, you'll know that the same factors are used multiple times. And the reason this can happen is because of what I've just told you, because as the cells progress from one state to the next, all the options that they did have been closed off, new options are available, and they can use the same factors now to distribute the decision-making across those options. And one of the ways the cells during the embryogenesis can manage to do that is they can do it through, for example, migration. They can move from one place to another where the same array of growth factors or inhibitors might be available. Positional information is important, and this will come perhaps towards the end of the talk when I talk about the blood differentiation protocols. And I'll give you some examples of developing systems from embryonic stem cells to illustrate how they're fundamentally self-organising. So this is true for a lot of cell types, and this idea underpins the organoid technology that people are using currently. As a last point, cells are not embryos, that biology doesn't always scale, and so if you're trying to come up with a treatment using a mouse model, it can be difficult if you're trying to translate that into a human. And of course, people are not mice. So first decisions are critical, and cells proceed stepwise through obligate intermediates. Okay. So this is, in a sense, a schematic showing you the first decisions that we deal with in the lab. Do you make mesoderm, which generates cells such as the gut or the blood or the heart, or do you make neurectoderm, which generates neural type tissues, the skin, and the head mesoderm? To do that in the lab, we activate TGF-beta signalling to get mesoderm. Or if we want to get ectoderm, we block TGF-beta signalling. But it's, as I mentioned earlier, it's actually a little more nuanced than that these days. What we would do now, as we proceed to the next step, is if we're wanting to generate mesoderm, we would activate BMP-type signalling and inhibit activin-like signalling, or vice versa. And under those conditions, in both cases, we're looking to try and push cells towards one step or another, way or another. So as I mentioned, we used to use, we have generated a lot of reporter cells, and this is one of the first ones that we generated. It's a GFP knock-in into a gene called MIXL1, which marks the primitive streak in the mouse and most vertebrate embryos. And what you can see is an embryoid body here. It's making mesoderm on the outside, which is fluorescing green. That green material will correspond to this green stripe here on this aerial view of a human embryo, which is essentially a disc. You can see there's a few things marked on that embryo. There's an epiblast. The cells that go on to make the new ectoderm and that head mesoderm will remain in the epiblast, whereas the cells that go on to make the mesendoderm derivatives, the heart, the blood, the gut, will migrate through this structure called the primitive streak. And during that migration, those cells will see either high activin signalling or high BMP signalling, and those combinations of factors will, to some extent, dictate the kinds of cells that emerge from the streak as the cells crawl out. So what this means in terms of practicalities is you can imagine a gradient of factors running along the length of that streak. Cells entering at the head end or the node end are seeing high activin signals, and they will go on to make things like endoderm and notochord. Cells seeing a little bit less activin and a little bit more BMP might make muscle. Heart would come out in the middle here, and at the very high concentrations of BMP and lower activin signalling would generate blood. So that idea has really come mainly from experiments in the frog, and we found it to be generally true for our differentiation experiments. So we can kind of recapitulate that idea here. We can set up a 96-well tray, and we can titrate factors, BMP across one axis and activin across another axis, and we can put our spin embryoid bodies into each one of those wells, and then it can look like this. So that's the MIXL1 cell line. So that's a photograph of a 96-well tray. Each green thing in there is an embryoid body. You can see some are greener than others, and that's because the MIXL expression is higher in those. You can also see how the factors affect the viability of the cells, depending on what you're looking at. So, for example, here, you've got high MIXL expression at this particular cross-titration point. Those cells will probably go on to form endoderm. And you can also see over here high levels of BMP, which are toxic. We can analyse that kind of thing by flow cytometry, and each little box there is a summary of flow cytometry readout. And maybe if I just get you to look at the day 4 analysis, you can see on the left-hand side here where you've got lower BMP and higher activin. You've got higher expression of EpCAM, which is an endoderm marker at this stage, and higher expression of MIXL as well towards the higher levels of activin. And on the other side here, you've got lower EpCAM, and you can see over here those two situations are reversed, where you've got high PDGF receptor and low EpCAM expression, and that's because those cells are heading towards mesoderm. So the first idea there is that gradients that happen in the embryo can be used to try and push cells towards different lineages. And on this next slide, what I've got here is a kind of cartoony summary of those concepts. I've got 3 different cell types, thymus, lung and pancreas. I've got 3 different green things there, which are reporters that we've made in the lab that identify FOXN1, a thymic specific gene in the endoderm, NKX2.1, which marks lung, and PDX1, which marks pancreas. And our heat map analysis of those reporter lines has led us a number of years ago to come up with a scheme, which I'll show you on this thing. And that is, you can imagine here a set of coordinates for differentiation towards particular lineages. In this case, the thymus requiring more activin than the lung. And as you move down that, you can see, again, different cell types coming out with different combinations of factors. Now, of course, this is a gross oversimplification of the whole process, but it gives you an idea that by adjusting the levels of morphogens in those very early stages of differentiation, you can dictate to a degree what kind of cell types are generated. Okay, so the cells, as they differentiate, proceed stepwise through obligate intermediates, and the binary decisions we've kind of covered slightly already. So in the next series of slides, I'll just take you through some of the work we've done with our pancreatic differentiations, I think. Let's have a look here. And maybe I can go back one here, because I think I've forgotten to point out that, as I noted earlier, the growth factors are used over and over again during the differentiation time course. The other key factor, and this is why the reporters work, is that the differentiating cells express the same genes as similar cells from the embryo at similar stages of development. So if I see PDX1 coming on in the culture, I can be pretty certain that those cells are heading towards a pancreatic fate. Under the right conditions, if I see FOXN1 or NKX2.1, I know that we're heading towards thymus or heading towards lung, for example. So in the case of pancreatic development, people have spent a lot of time thinking about this, and that's because what the field has been after is generating insulin-producing beta cells that could be used for the treatment of type 1 diabetes. So you should be aware that type 1 is caused by destruction of the insulin-producing beta cells in the pancreas, and you might be aware that that can be treated quite well with an islet transplant, but the problem is, of course, that no one can hand over their islets because you just can't get them out of a living person without causing severe damage. So all of the islets that people get transplanted are from deceased donors, and there is a massive shortage of those. So if you could generate that cell type in the lab from pluripotent stem cells, then you could solve a really big problem. So people have thought about this a lot. And so if you look at the progression of cells as they move from what is essentially a pluripotent stem cell, which is vaguely in this area here between inner cell mass and epiblast, those cells express genes that similar cells in the embryo express, such as OCT4. In the lab, we tend to use CD9 and EpCAM because we can run them on a fax. If you push those cells with nodal signalling, so that means activin in reality because they deliver the same kind of signalling, and Wnt signalling, then you can push them towards definitive endoderm, and you can tell that's happening because they'll express things like FOXA2 and SOX17. Not KDR, that's a mistake there, ignore that. And then in the next step, if you treat those cultures with FGF, you can posteriorize the foregut, and those cells will now be expressing PDX1, indicating they're starting to become pancreatic. And using inhibitors and activators of TGF, beta and RA signalling, you can then push them to the next step. In most protocols these days have a final maturation step, which involves things like thyroid hormone. And eventually you end up with a spectrum of cell types that are expressing insulin, glucagon and somatostatin. And I guess sort of taking you through this process again, what people have always found when they go through these kinds of steps is that every time you do something and generate a new cell type, then there are more options that the cells have available to them. So in the original protocols, people tended to focus on these factors underneath here, where you could push a cell towards, say, endoderm with activin and then towards pancreatic endoderm with retinoic acid. But the newer protocols, as I mentioned now, actively block the alternative pathways to try and enrich for the cell types they're interested in. And of course, one of the reasons people like us rely on developmental biology, as unfundable as it is in the context of NH and MRC, is because it gives you a clue as to what kinds of factors you need to add to your cells to push them one direction or another. Okay, let's have a look at this. So I don't have many data slides. This is quite an old one. These green balls of cells are insulin-positive cells. They're green because we inserted GFP into the insulin locus of a human ES cell line. We differentiated them for about 30 days along that kind of pathway, and then we sorted them because we could, and we reaggregated them into little balls that, you know, you could probably convince a funding body was an islet. What we did observe, though, is that once you'd sorted on GFP, over time, those cells gradually lost their identity. And you can see on this lower panel here, they've gone on to express things like glucagon, and the only remaining insulin-positive cells are expressing NK6.1, which is a marker of a genuine beta-type cell. So this is another example of cells reaching a decision point. They've got there, and now they've got a whole lot of other options available to them. And in that case, we were not able to find a condition that held them in the insulin-positive state. There are other reasons for that, but that's an example of, in a sense, the way your cultures dictate what cells you generate. And again, this is why you can't really infer human development from these kinds of systems. So cells are wired to execute their normal functions, and that is they do want to differentiate, and as long as you give them some vague instructions, they will quite happily differentiate into a whole range of different cell types. So I think, what have I got coming up? Oh yes, okay. So the whole idea of organoids, which you will have seen is very popular in the literature at the moment, is something that people in the stem cell field have been grappling with for a long time. Originally, it was something we didn't really want. It was something where you were trying to generate a specific cell type, you set up your differentiation, and you generated this blob that only had a small percentage of cells you were interested in. In those days, that was definitely a no-no, but fashion changes, and so currently it's much more desirable, or often desirable, to generate mixtures or blobs containing semi-organised structures that resemble partly the organ that cell type resides in. So here's our little embryo map again. I'm going to show you a picture of our NKX2.1 reporter, which is a lung marker under these conditions. This is actual data off a heat map experiment where we've titrated the BMP and activin over those first 4 days. And the red area is where we got the most frequent appearance of green cells. So that's technically quite simple to understand. You can look down the microscope, you can score those visually, but we tended to run them on the FACS to quantify that. And you can see what a FACS plot looks like out of that area. What we did is we took those early clusters of cells at around day 4 of differentiation and transplanted them into immunocompromised mouse under the kidney capsule. And what you'll see here is that after a number of months now, what had happened is that those cells had continued to grow and they differentiated to form these green tube structures. We hadn't given them any instructions. All we'd done is, for 4 days, give them a particular combination of BMP and activin. And if you look histologically through that material, you can see that these cells have gone on to generate a whole bunch of different respiratory epithelial cell types. They've even gone on to generate structures that have smooth muscle around them in the same way you might find with an actual genuine piece of lung. And so And on the immuno here, you can see the NKX2.1 expression on the epithelial lining there. So I included that particular slide there just to show you as an example of what those basic instructions at the start of the differentiation could lead to in terms of a differentiation outcome with the cells self-organising. So I think in the last part of the talk, I'm going to tell you about some of the stuff we do in the blood area, and hopefully this will highlight some other areas about the complexities of trying to get particular kinds of cell types. Most of Andrew Elefanti's research over the last 20 years has been to try and make a haemopoietic stem cell. And this is useful because, as you know, your haemopoietic stem cells are the cells that can make most of the cell types circulating in your blood in the adult. And if you could make them from a pluripotent stem cell, then rather than having to get a bone marrow transplant in cases where you had, for example, a leukemia and needed a new blood system, rather than looking for a match donor, you could potentially take a skin cell, turn that into a pluripotent stem cell, and then turn that into a haemopoietic stem cell. And that would be a perfectly matched haemopoietic stem cell. It's good in theory, but it's obviously trickier than that, and people haven't been able to do it. And it's trickier because the development of the blood system is not so straightforward as that first picture I showed you with a haemopoietic stem cell at the top making a whole bunch of stuff. The way the embryo operates is that it only makes the stuff it needs when it needs it. So it doesn't make a haemopoietic stem cell and then make a blood system. The first thing it needs is red cells, so it makes those first. Maybe it needs some macrophages or myeloid cells to go and clean up all the mess, so it'll make those next. Eventually, maybe it'll need some kind of immune system, so it starts making things like T cells and B cells. Only once it's done all of that does it make a haemopoietic stem cell. And so in distinction to the way the adult system works, where you have a stem cell at the top making derivatives, during embryogenesis, the blood system is built from the base up. And what that means is that the appearance of blood in your cultures does not mean you've got a haemopoietic stem cell. It means you've got blood. So this little scheme here shows you some of the developmental milestones that happen during human blood development. There's a days across the bottom there. And I should say, one of the things I didn't put on my slides is that cells differentiate at a certain rate, and although you can speed things up by pushing them through decisions as quickly as you can, there is a certain clock speed that they run at, and it's not always possible to speed things up faster than they occur in the embryo. So necessarily, experiments with human ES cells take a lot longer than they do with mouse cells. The first blood is made in the yolk sac, and this, as I mentioned, is red cells, some megakaryocytes, and some monocyte precursors. Microglia are also made during those early phases of blood cell development, and lymphoid cells can be derived from those cells, although it's not clear whether in the embryo they're actually made there. What have we got here? The haemopoietic stem cells themselves come from this area here, which is the dorsal aorta, and on the ventral floor of that there's a cluster of cells that bud off. These are intra-arterial clusters. They will give rise to definitive haemopoiesis. Most of these are what are now called pre-HSCs, so they're not even haemopoietic stem cells at this stage. They're cells that, under the right conditions, can become haemopoietic stem cells. They're rare, long-term repopulating cells. Those clusters also generate a whole spectrum of other precursors that are not stem cells. They generate the myeloid precursors, they generate lymphoid precursors. And so all of those cell types are in there to try and confuse you as you try and generate your haemopoietic stem cells. So the discovery, and I think the paper that I asked people to have a look at, and as part of this seminar, was one we published a couple of years ago, where what Andrew and Elizabeth identified was that signature difference between the cells you make in the dish versus the cells made in a real embryo could be distilled down to the expression of a particular bunches of HOX genes. So HOX genes are essentially genes that dictate the axial position of cells along the embryo, and that happens in flies, and it happens also in vertebrates. And what they observed is that the cord blood cells, which are marked CB here on the heat map, expressed a whole range of HOXA genes, whereas the cells that were made in the lab essentially didn't express those. So that meant at least we had something to go looking for. We'll go looking for conditions that elevate HOX gene expression. But where do you go looking and when do you go looking? So this is our little scheme of haematopoietic stem cells that we'd see in the adult. And then the annoying thing is that to get there, we have to go all the way back to the beginning. So as I mentioned, those first decisions that you make in the first 24 hours send you on the trajectory you want or don't want. And so the interventions that we ended up looking at, before I say that, the other thing I'll show you here is that I think might be, yeah, so let's see, is that going to... Right, well, technologically challenged. Apart from the two forms of hemogenic endothelium I'm showing you there, which is, let's see, can I get that arrow over here? Yeah, so we have this yolk sac-like hemogenic endothelium, which you can see can generate a whole range of different cell types. It can generate myeloid and erythroid cells, it can generate T cells, it can even generate certain kinds of B cells. The only distinction between these two arms is that on this side of the equation, we've got cells that are expressing the HOXA cluster, and on this side, they're not. I'll also point out this little side group here that is from the various early stages of blood cell differentiation. And in this case here, the mesoderm effectively gives rise directly to these cell types you see here, the erythroid cells and the myeloid cells. So that's the distinction. And what Andrew and Elizabeth found is that if they could treat cells with a short pulse of TGF-beta and to block TGF-beta signalling and to activate the Wnt pathway, in this window here between days 2 and 4, for approximately 2 days, they could activate the HOXA genes, and that would divert cells, rather than going towards this primitive pathway, going towards this definitive pathway. And what that looked like in culture, using those reporter cells, is apart from generating blood, which we would have got out of the primitive pathway on the left-hand side, you've got these vascular structures which were not present in cells that had not seen that particular treatment. And I guess one of the things I'd remind people here is that the difficulty with this kind of approach is that we have to guess the concentrations and the timings of all these factors. I mean, you have a rough idea, but essentially it's an experimental process. Okay, and you'll see here a couple of boxes where you've got endothelial tubes with green cells budding off from inside them. So using that method, we were able to go on and characterise the cells that came out of that method. And what we found is that, by and large, these cells had a very similar transcriptional profile to cells that came out of human AGM. So we were very fortunate to have collaborators that were able to isolate the AGM, so that's that dorsal aorta area where the clusters are coming out, from human embryos and to sort fractions. And you'll see here, particularly if you're looking at the cells that are sorted from the AGM versus this group of cells over here in the second set of columns, the very early endothelial cells, which are endothelial marked as EN there, or the stem progenitor cells, have a reasonably close similarity in terms of gene expression profile. And this was true when we went and looked at other things such as transcription factors and signalling pathways. And I think the thing that we also observed is there was an activation of the HOXA cluster under those conditions. I don't think I've got that slide in here, but it wasn't complete. In other words, there was still more work to be done in terms of matching that profile up with what we were seeing in the embryos. Okay, so just to show you what those two cell types look like now in comparison. On the left-hand side here, you've got the primitive differentiation protocol. You see a whole bunch of blood cells here in the slide. You can see these patches of red endothelial cells, which are haemogenic, but they're not organised. Simply that 2-day pulse of TGF-beta inhibition and Wnt activation dramatically changes the cultures. And you can see here in this picture on the right here, the vascular structures. Okay, what else have I got? So at the moment, we can't generate haematopoietic stem cells from our pluripotent stem cells, and we've got a long way to go. As I mentioned, we're fiddling with all of the conditions at this stage, and even so down this stage here to see whether there's anything we can do to our cultures to push them towards the cell type we're interested in. But in the meantime, what you should have also gathered is that we can make actually all the other blood cell types. So we can make monocytes and macrophages and dendritic cells. And I'll just show you briefly some of that in the last few slides. And we can make T cells and B cells. So all of those cells, from our point of view, are useful cell types for example, we want to be able to match them to the cell type that you're actually putting in there and be aware that that cell type is going to have to know what to do with it.
Thank you for the interesting talk. So you had a diagram where you had a ball of cells and some of the cells were felt to be held in an insulin positive state. Mm-hmm. And there were different types of cells in there. And my question is, if all of the cells were subjected to the same conditions and the same factors, why it ended up with different cells types?
Well, that's actually quite an easy question to answer if you think about it, because the cells are not islands, they've got neighbours. Okay, just like the cells in your tissues have neighbours. So although we can put a growth factor onto a culture, they're all stuck together. And so one of those cells is going to go, hey, I've just seen this growth factor, what do you think of that? Immediately, the cell next to it is getting two bits of information. It's getting the growth factor you put on and the other cell telling it, what do you think of that? And so that whole process goes through the cultures all the time where they're talking to each other. And so they're continually trying to rationalise what you're telling them to do, which is the uniform defined media, and what they're telling each other to do. And so you could appreciate that in a developing system, if you're trying to generate a particular structure from a particular precursor pool, those cells aren't all going to do the same thing. Some are going to do one thing, and they're going to tell the other cells, I'm doing this, so don't you do that, you do something else. And that happens all the time.
Are positionally related genes like HOX genes, are they like a marker that these cells have gone down a pathway which is suitable for developing like blood cells, for instance, or are they also feedback and influencing that development?
So could you just repeat that? I didn't quite catch it.
Are positional genes like HOX genes, which are like expressed differentially along the body, are they influencing, say, like the development of something in the blood cells, or are they just marking that it's in that?
So are they actually physically involved in the decision-making process about what you become, or are they simply a marker of the path you've taken? Is that the question?
Yes.
Yeah, so We actually don't know the answer to that. Part of the... Experimentally, people have tried to overexpress some of those HOX genes that are a definitive sort of signature, and they've been largely unsuccessful in pushing a cell into a different state. That is complicated though, because a lot of homeobox genes, you might appreciate, are transforming, so they tend to make cells grow. And one of the characteristics of a blood system that is containing a haematopoietic stem cell is that it grows, can repopulate an animal. But then trying to figure out, is it repopulating the animal because you've transformed it, made it malignant, or is it because it's correctly patterned, becomes a difficult thing to figure out. So at the moment, we don't know the answer to your question, is it simply an address? If it is, that's fine, as long as we know what that address is. Or is it actually part of the wiring of those cells about what they are?
So you said that the differentiation decisions are over in the first 24 hours, which restricted to some village. Was that surprising? Because in particular in human development, to go from the inner cell mass to something that is that restricted is a bit longer.
Yeah. So what people have found probably over the last 15 years is that as they do more differentiations, they just things get faster. And the original protocols we would have used in the old days to get up to that mesoderm stage would have taken 6 or 7 days. But people have figured out how to sort of short circuit that so it happens now within 24 hours. Now, in the embryo, you're right, that process would be much longer. And what we think is that a lot of the timing in the human embryo has to do with organisation. So you could proceed from step A to step B very quickly, but you have to put your cells in the right place. You have to develop structure. That developmental process looks like it can be uncoupled from the differentiation process. So it means that you can push cells to differentiation from one state to another very quickly. That just requires changes in gene expression. But obviously in that process, they're not developing a structure and organising themselves. So I think that's probably the basis of that difference.
I think we should leave it there, but for the students who are interested in speaking to Ed further, we're going to have luncheon today just because of all the strategic stuff that's going on. It's going to be in the tea room. We've got a table reserved out there if anyone would like to join us. Can we just have a break?